Hello and a warm very welcome everyone to our today's webinar on bioanalytical aspect of BEB studies for endogenous molecule. Myself, Hemal Kurani from the Bioanalytical Department, Senior Group Leader and represent the Veeda Clinical Research Limited. Veeda Clinical Research Limited together with its subsidiary Bionits India Private Limited as well as its joint venture Ingenuity Bioscience Private Limited, together referred to as Veeda Group, which offers a comprehensive portfolio of clinical, preclinical, as well as bioanalytical service to support innovator, biosimilar, and generic drug development program of our global client. Together, we serve clients globally in the following industries like pharmaceutical and biopharmaceutical, agrochemical and industrial chemical, herbal, as well as medical device. Now, as your facilitator today, I would like to introduce you all of a couple of points as we begin this session. This session will have a presentation in the beginning, followed by the panel discussions. We will be running a live question and answer during the webinar, which will be led by our backend team present with us there. So for all your questions and queries, we have enabled our ask button features. It's right on your screen, so you can drop your questions in there. Now, I would like to introduce to our presenter for today's discussion, Dr. Pritesh Contractor. Dr. Pritesh Contractor is presently working as a principal scientist in Bioanalytical Research Department at Veeda Clinical Research Limited, where his role is to serve as scientific and technical lead for biopharmaceutical companies. He plays a key role in the development, validation, as well as application of bioanalytical assay to comply with scientific and regulatory requirements. Dr. Pritesh Contractor has over 16 plus years of professional research experience of regulated bioanalysis in developing, validating, as well as applying bioanalytical method to support clinical studies of generic product as well as new chemical entities. He has experience on developing method of endogenous molecules, prominently 25 amino acid, chiral molecules, generic product by LCMS-MS technique. He has also experience on developing elemental analysis, like it means various ion formulations such as ion sucrose, ion dextran, ferric carboxymaltose, then potassium chloride, zinc sulfate extra by ICP-OES techniques, when the element is derived from the pharmaceutical being administered. Now, I would like to call Dr. Pritesh Contractor for presenting webinar on bioanalytical aspect of BEB studies for endogenous molecule. Dr. Pritesh Contractor, over to you. Thank you.
Thank you so much, Hemal, for your kind introduction. And I would like to welcome to all our participants to join these presentations. And I'm sure this presentation is surely useful for all of you. And definitely we can discuss many things from these presentations. So as mentioned by Mr. Hamel, that today's our topic is for this bioanalytical aspects of BAB studies for endogenous molecule. I hope that slides is visible to everyone. Once Hamel, can you please confirm?
Yes, Dr. Pritesh Contractor, it is fine with us. You can carry on.
Yeah, so I'm sure everything is fine. The communication is established. So the today's topic is the bioanalytical aspects of the BAB studies for endogenous molecule. In today's talk, we are going to discuss the following index I just mentioned on my screen is the first of all the definition of endogenous molecule, then BAB study execution challenges. What two types? These are the main two challenges we observe during the execution of study. The first one is the endogenous level cannot be distinguished from the exogenous compound, and another one is the diurnal variation of endogenous molecule. This is also the issues observed during the execution of study. In with respect to bioanalytical concern, the selection of analyte-free matrix for the preparation of CCs and QCs, it is also a challenge how to select this matrix. And then finally, the quantitations of endogenous drug in human matrix, how to quantitate when we are using some alter and some different matrices, how to execute that analysis, that we can discuss in the subsequent slides. Then we can discuss about the validation requirement, what regulatory says about the validations when we are performing validation for the endogenous compounds. And then we also discussed the case studies, specifically that's case studies. We already did the method development and validation at Veeda, and we have a very good experience with those molecules that we discuss at the end moment. And the finally, we can discuss the Veda's experience and expertise, how many molecules we did completed successfully. So that all parts we conclude in these presentations. So now we can start with the first part, definition. So how do we define the endogenous molecule? So the endogenous molecule are those that originate from within a system, such as an organism's tissues or cell. The endogenous level may vary because of age, gender, diurnal variations, illness of as a side effect of drug treatment. So it is already present in the body, and that's why it's called endogenous, means inside the body or caused by factors within the body. And following are the very known examples, and we have categorized in the different categories. The first one is the hormones. The hormones are the thyroid hormones. That's an example is the levothyroxine. Then the sex hormones, the estrogens, progesterones. Vitamins, then vitamin D, vitamin K, thiamine, that's vitamin B. When we talk about the ions, then potassium chloride, that will measure the potassium. Then iron in iron formulations, amino acids, that isoleucine, leucine, valine, and many more. In our body, there is a more than 20 amino acids are present, and which are essential for our functions of various body mechanisms. Then the bile acids, ursodyls, then the omega-3 fatty acids. So these are the categories of endogenous molecule where we perform the BAB studies routinely. And these molecules are required to well define and well characterize and during development, during validation. Now, as mentioned, there is a two major challenges for study design and that bioanalytical issues what we face during the method development. In normal drug, what happened, the pharmaceuticals companies that develop generic versions of those products, tablet, capsules, and IVs, are typically required to demonstrate bioequivalence to an approved reference product. At the same time, assessment of the bioequivalence of a certain generic and the reference compound is complicated by the presence of endogenous level of said compound. So that complication must be resolved during the execution part. So here we discuss the first challenge during the study design, the presence of endogenous level of said compound, which cannot be distinguished from the exogenous compound level and following the drug administration. So how to distinguish, because it is already present endogenously and we also administer from the exogenous. Then when we measure the total compound, then how to distinguish these two things. So here is the resolutions, which is very much important, that estimation of specific biomarkers, which provides the measurement of endogenous analyte of interest by using the established procedure. One of the best and the elite example is the estimation of insulin aspart using the C-peptide as a biomarker from the serum insulin. So these methods we have already developed and validated at Veeda, and we have a very good results, and we imply to the study sample analysis also. So this is the best example for this challenge. So the studies were conducted in healthy subjects. It was necessary to correct the serum immune reactive insulin aspart concentration for endogenous insulins. So what we did in this, each subject had a blood samples taken for the measurement of serum C-peptide concentrations at the same time point at the PK sample. Means here the two separate method development and two separate method validation is required. The first one is the total insulin, which is giving the combinations of insulin aspart and the serum insulin. And the second method is developed and validated for the C-peptide from the serum. The two separate method is required, and the time points for both are remain same. Now, correction for endogenous insulin concentration using the C-peptide concentration data was performed based on the Owen's method using the following equation. So this equation is useful for the calculation of the insulin aspart concentration. So here the serum insulin concentration is used, one factor F is applied, and the C-peptide concentration is also used. Now, how to calculate the factor F? So where F is the average of the ratio of serum insulin to C-peptide at baseline, where the baseline was -60, -30 hour, and 0 minute for the study. So these baseline samples are very much important to calculate the factor F. Now, how the F factor is calculated, we'll see in the next slide. In this next slide, you can see that the first column, the equation is mentioned how the factor F is measured, the serum insulin at baseline divided by the C peptide at baseline. So in the table, you also see the zero pre-dose concentrations of serum insulin and C peptide. That's a 95.504 for serum insulin and 1245.706 is for the C peptide. Now this factor comes out is 0.0766. Now this as F factor is used throughout for the calculation of other subject samples concentrations. So here for the remaining time points. So the first one we give as an example that at 0.33 hour sample where the concentrations observed is 208.622. So you just see in this table, by using these two methods, we have all three data, the serum insulin data, we have a C-peptide data, and finally we have an insulin aspart data, which is our requirement for the study executions and for the final PK assessment. So this is one of the best approach to be applied for the measurement of such insulin, of such endogenous products. Now the second one is the diurnal variation effect of this endogenous molecule, because we know it's already endogenously present. So throughout the day, its level is ups and down. So typically for the steroid compounds, the all steroids with exceptions of estrone and estradiol had a diurnal variation with the significant within the subject variation. For cortisol, cortisone, pregnenolone, 7-hydroxypregnenolone, these are the steroids where the endogenous level is varies and the mean concentration at time interval in the early morning 8 o'clock just after awakening was significantly higher than the mean concentration in all other times interval. So in the next slide, we can see this is from the graph. In graph, you can see the time versus the concentration graph. And it is a very much clear from the graph that at the 8 hour in the morning, the concentration is quite higher compared to the other time intervals of the day. So these types of diurnal variation is also going to affect during the execution of the BAB studies. So that part should be also taken consider. So these are the variations. In the next slide, we can also see one exceptions. For all major steroid with the exceptions of allopregnanolone, the peak concentrations were observed around 8. Allopregnanolone, the peak throughout the day, its highest concentrations occur at around 12. So that is a little bit different. And I say in the slide, in estradiol and estrone, there is no major changes. It's a similar trend. So from this graph, at least we know how the diurnal variation is going to impact during the analysis. So that part also to be considered for its evaluation. Now, how to resolve this concern? So these are few good examples or some approaches that can be applied during the study or execution. First one is the modulation of the baseline endogenous level. Second one is the restrictions of the dietary intake of the relevant substance. And third one is the correction for baseline level of endogenous compound. This third one is widely used for all endogenous molecule, and other are the very rarely used, but the third one is widely used for all endogenous molecule. Use of matrices other than blood, plasma, or serum. So change in matrices can also give the very much improved results in with respect to the diurnal variation effect. We'll see the one by one. The first one is the modulations of the baseline endogenous level. This approach has been used to assess the bioequivalence of growth hormones, follicle stimulating hormones, and luteinizing hormones preparations. For example, somatostatin infused to healthy volunteers before recombinant growth hormones administration in each period of their crossover study. Then these somatostatins reduce its level, suppress the endogenous growth hormone level, although not to zero in all patients, but one such types of suppressions definitely observed in the endogenous level that can be further optimized and used during the pilot study execution. The second example is the dexamethasone. 4 mg dose will be administered 10 hours prior to study drug, that's the hydrocortisone administration as a pre-treatment to lower the endogenous hydrocortisone level. So such infusions of these supporting drugs gives you the modulations in the baseline and reduce the endogenous level. The second option is the restrictions of the dietary intake of the relevant substance. The best example for this given by the FDA, this guidance for the bioequivalence studies of KCl, potassium chloride. Supplements are good example for these approaches. Diet should be standardized to provide the following daily intake. So these figures are mentioned in the guidance that potassium quantity and amount is 50 to 60 milliequivalent, sodium is 160 to 180, and the overall calorie is also mentioned. Water administration is playing a big and the important role for this potassium analysis. So the water intake is definitely increased. In routine diet, the water intake is about only 1.3 to 2.5 liters per day. But during the study, during the baseline sample collection, it is increased to 3 to 5 liters per day. So by this restrictions of the dietary, the potassium studies can be executed. The second example is the bioequivalence studies of iron formulations. Iron sucrose, ferric carboxymaltose supplements this. So patient should not take the iron-rich food during the study period. Because what happens if the patient is taking the iron-rich food, in that case, if the patient is the anemic patient, so the iron level may be fluctuate, maybe varies if the subject or patient taking the high iron rich food. The few of the examples are iron rich food are this: the meat and eggs, bread and cereals, wheat products, seafood, vegetables, specifically the spinach. Spinach is a rich source of iron. So if the study is conducted on patient, so the patients are prohibited to take these iron rich food, and this is very much required. Otherwise, if they take these types of food during the study, then the iron level is definitely goes higher and will see the spikes in the concentration profile. So by this these restrictions of dietary is very much important for the endogenous compounds, and that can be also worked out during the study execution. Now, the third, which is the widely used approach for all studies, that the baseline corrections. The most frequent correction method is simply to subtract the baseline endogenous compound level from all subsequent post-treatment value. The baseline level is usually taken as the mean of 3 or so level in the 24-hour prior to dosing. So we all aware that the phytonadione, that's vitamin K, iron, potassium, levothyroxine, the normal endogenous level is mentioned in this table, and at the other side we mention the baseline sample, the time points, how it is collected. In phytonadione there is about 7 to 8 pre-dose samples, then in iron there is a 3 pre-dose samples, in potassium the maximum there is a completely 3 days required for the baseline equilibrium and the for baseline sample collection. That's a totally different study design for the potassium. And for levothyroxine it is a minus 0.5, 0.25 and 0 hour. So by this way, based on the available endogenous level, you can select the time points for baseline corrections. The fourth one is use of the matrix other than the blood, plasma or serum. So now what happened in the endogenous molecules, the homeostatics mechanisms maintain the level of an endogenous substance within a physiological range and prevent change in the substance level that may be the harmful. The example of the potassium chloride. Now potassium measurement is performing by using the urine sample, not in the plasma and not in the serum. Why? Because the potassium is the electrolytes, and that is not giving a higher difference in the plasma or serum, because there is some equilibrations with the cell and the plasma. So in the cell, the potassium level is about 150 to 160 milliequivalent, with compared to that in plasmas, their level is very less, about 10 times, or it's only 3.5 to 5 milliequivalents only. And due to the active ion transportation, this equilibrium is constant in the plasma and serum. So when the product is administered, then there is no much higher variation you observe in the plasma or serum. So the best example to measure the concentration in the urine. because in a steady state, potassium absorbed by the GI tract is equivalent to the potassium excreted in the urine. So by the urine potassium measure from the urine, you will get the best and the consistent data. Similarly, when you are targeting the iron formulation, so you cannot use the blood because iron is the Fe, and from blood there is a uh, mm, red blood cells and everything, which significantly impacted on the estimation of the iron. If similarly, if you use the plasma, there the anticoagulant is mixed, and when anticoagulant is mixed, then premix is required. And during the premix, there is a maximum chances of the hemolysis of the plasma, and the hemolysis nature of that plasma is significantly affected on the iron estimation. So therefore, the serum is considered for the estimation of the iron. So these types of changing matrix can also give the improved result for this endogenous molecules. So now the one part is completed, that what are the challenge for the clinical point of view. Now we are going to discuss the bioanalytical challenges. In bioanalytical, when we are targeting to develop the method for endogenous, then first questions comes in the analyst mind is how to get the analyte free matrix, because it is endogenously present, then how to get the analyte free matrix. So these are few procedures that we can use to get the analyte free matrix. Here the some options are mentioned. First one is the usage of the stripped matrices. Second one is the usage of surrogate matrix. Third one is the blank subtractions. Fourth one is the standard addition method SM. Let's see one by one how this method is useful to get the analyte free matrix. First one is the charcoal. Charcoal is well used and widely used for such molecules. Charcoal is an amorphous form of a carbon and is present as a powder, and it's highly porous in nature. So an adequate amount of charcoal should be added to the plasma or serum, which absorb and remove the intended analyte from matrix and make a matrix analyte free. The treatments includes a uniform vortexing for specific period and followed by a precise centrifugations and filtration to avoid such interference and some clotting. So that's why this precise procedure is required. This approach is successfully applies for the estrone, estradiol, levothyroxine, hydrocortisone, thiamine for the preparations of the analyte-free matrix. So these are methods we have validated at Veeda and successfully used for the analysis also. Now the second approach is the photodegradation. This is something new and something very creative. The physicochemical properties can be used for the degradation of photosensitive molecule. Expose the plasma or serum to direct sunlight for its gradually degradations with the respect to specific time duration. After that, matrix should be evaluated for its possible presence and adequacy. It means a screening, that we need to screen the plasma and see its available level. Now the phytonadione, the vitamin K, is one of the elite example of this approach, where photodegradation is used to obtain the analyte-free matrix. This is one of the very creative and very light method of our Veeda, that we are using this photodegradation method for the phytonadione to prepare the analyte-free matrix, and it's excellently worked. And we did so many studies of phytonadione with this method also. Now, the next one is the usage of the extraction cartridges for filtrations of matrix. This is also widely used approach. Sample matrix is applied to a SP column containing a sorbent with selectivity such that it will retain the analyte of interest, and the filtrate provide the analyte-free matrix. Examples include anion exchange, cation exchange, and mixed mode sorbents. This technique successfully applies to L-carnitine. It's one of the amino acid and N-acetylcysteine to remove the analyte from plasma by using the SP column. The sorbent filled with the amine NH2 group used for the retention of this analyte. So as per the SP principles and as per the SP protocol, you can select the relevant SP cartridges to retain the intended drug inside the cartridges, and definitely you will get the analyte-free matrix. Now the next one is the usage of the surrogate matrix. Being a surrogate matrix buffers, phosphate buffer, saline, then 2% bovine serum, normal saline, milky water are widely used as an analyte-free matrix. When the stripped matrix are not providing analyte-free matrix, then usage of surrogate matrix is one of the widely accepted approach for the quantifications of endogenous molecule. So because why we are using this surrogate matrix, if the other about three options are not useful and not providing us a clear plasma or clear matrix with respect to our LLOQ, then we have to use this surrogate matrix option. Few molecules have high level of endogenous amount in respective matrices are difficult to remove from authentic matrix or true matrix. Matrix like aqueous humor is rare and difficult to obtain, then surrogate matrix can be used for method validation purpose. Here the surrogate matrix can also be used in a rare matrix because we all know that aqueous humor, it is a very less quantity available and you cannot procure it as a routine like a plasma and serum. Therefore, if we are doing a development of any specific molecule and the matrix is the aqueous humor, in such cases, we have to plan to plot our calibration curve by using the surrogate matrix only. Milky water used as a surrogate matrix example, the potassium in urine matrix and the few amino acids. However, it is very much important to observe the matrix effect during the usage of the surrogate matrix. Yes, this is one of the very important point to be considered during the method development because normally not all surrogate matrix are compatible with respect to the response of ISTD. If there is a major changes, then you will see the significant response variation in the ISTD. Your ISTD response in a surrogate matrix and the ISTD response in a true or authentic matrix is significantly vary, then your method is definitely not working and it is going to be failure. So that part is very much important to observe and to optimize during the method development stage. Now the blank subtraction approach, when above two options, stripped matrices and the surrogate matrix not effectively working, then blank subtractions can be applied to remove the endogenous amount of analyte from the calibration curve. Yes, because there are sometimes it happens the stripping is not working and that surrogate matrix is also not working because of the higher matrix separations and enhancement. So in that case, the similar matrix can be used for the plotting of the calibration curve. But in that way, you have to do the blank subtractions and accordingly you can plot the zero through calibration curve. But in this method also there are some limitations in that you have to fix your LLOQ accordingly because whatever the endogenous concentration is available. This method is only applies if the LLOQ level is equivalent to half of the possible endogenous content of used matrix. The method is successfully applies to quantifications of total serum iron with LLOQ is 0.5 microgram. Now see in this slide you can see the 3 replicates of blank is taken and the mean is calculated. And now this mean value 1, 2, 3, 3, 4 is subtracted from the net response of the CC standards. And finally corrected response is calculated here in the last column. And now this final corrected response is used for the calibrations of the standard. So here the final output after the subtraction, you see the accuracy is about 95 to 100. And you see the slope value, intercept value, and the R value. So accordingly, this blank subtraction method is used for the calibration curve when we are using the same matrix for the calibration curve. Now, bioanalytical challenge is the second part. Now, the standard addition method. This method is no more practically too much in use, but there are some used in few of the molecule, but it's not like because at this moment we have a ISTD available of deuterated or maybe the analog, so that cannot be exactly practically applicable, but still I explain in this session. The standard addition method is recommended when no internal standard are available to compensate for pronounced matrix effect. In particular, in multi-component analysis, finding an adequate IS for each analyte is not always possible. Therefore, the standard addition method comprises the addition of a reference standard of the analyte of interest at increasing concentration level into the multiple aliquots derived from the same biological sample in which the endogenous concentration of the analyte has to be determined. And one aliquot remain unspec, which is equal to the blank, which we discussed in the earlier slide. So after that, what happened? After analysis, the detector response is plotted against the added concentration and the endogenous concentration is derived from the negative intersections with the x-axis. So whatever the endogenous concentrations observed, that can be further identified by extrapolating the graph and with these intersections. So this is the provision of the standard addition method. Now the second one is once you fix your analyte free matrix that is selected, then how you can quantitate the methods and how that analyte and everything. So here the procedure is mentioned. Uh, Process standard 0 samples in replicate of 3 or 6 and analyze along with the calibration curve. Here standard 0 is the blank sample. There is no drug spike, only the ISTD spike in this sample. This standard 0 sample is a process filled with respective ISTD. Based on area of endogenous content of particular matrix lot, calculated concentration should be evaluated. Mean value of the standard 0 should be further used for the amendment of QC samples. Evaluations and acceptance of QC should be based on the final amended concentration, not from the nominal. Below is result table represents how this QC's amended concentrations derive. So here we can see the nominal concentration of HQC is 190 and LQC is 150. So after amendment of EC, the final concentration is 194 and 5.90 something. It means the amendment value on the EC value is about 4 point something. So how it is amended, we'll see in the next slide. See this result table will see everything. In the sample name column, you can see there is a 3 sample. That's a standard 0 EC used for to identify the endogenous level. In the calculated concentration column, second last, you can see the value of EC 4.8, 4.0 and 4.2. So now these three replicates mean is considered, and that is amended in the HQC concentration. So now analyte concentration, the first encircle HQC quality control sample, you see the concentration is 190, 95, 15 and 1 point. That's the nominal concentration. Now at the bottom, there is the amendment concentration. You can see that's a 194.401 and 5.901. And that is amended, added with the 4.2 or 4.8, whatever the mean coming from the EC. And you see the final last column, the accuracy of all quality control samples with this approach. You can see the perfect accuracy with this amendment approach. So this is the best approach to calculate the quality control samples in the true or authentic matrix by using the EC value. Now, what are the regulatory requirements for method validation? So being an endogenous presence of intent and molecule in human matrix, the experiment design of few experiments are different than normal method validation approach. The procedure should be predefined in method validation plan with appropriate rational and scientific justification based on the pre-method validation data. Because when the molecule is endogenous, definitely we cannot perform everything as per the normal methodology. So these are the few list of experiment. This is significantly changed when you are going to do the validation of endogenous compound. There are many more normal experiment also, but in presentations I have included only the specific experiment which is going to be changed for endogenous molecule. First one is the specificity. Second one is the precision and accuracy, specifically for the altered QCs. We are using the stripped or surrogate matrix, so the QCs prepared for them. So for that is required. Recovery and matrix effect. When you are using this different matrix, then how to measure the recovery and matrix effect. Stability experiment in authentic and altered matrix. The both stability is required, not only one, but we need to perform both the stability experiment and the dilution integrity experiment also, because we are using the different matrices. So how to perform the dilution integrity experiment. Now, these are few experimental details mentioned in the guidance. The first one is the ICH M10, then USFDA and EMEA. So preparation of CC standards, as we discussed in our presentation, they also mention how to prepare the analyte-free matrix and its rational and scientific requirements, selectivity, recovery, matrix effect. One new parameter is introduced, which is the parallelism that we are explained in the last or few after subsequent slides. Accuracy and precision, and stability in authentic matrix and altered matrix. So these are the basic details mentioned in the guidance also. Now, first one is the specificity. Due to endogenous presence of molecule in matrix, the specificity experiment for authentic matrix or true matrix is not possible because it's endogenous presence. So what we do, first we need to perform the specificity for internal standard in that authentic matrix. So that is required to observe any possible interference at the RT of IS in the authentic matrix. Now, when we are using the alter or strip matrix, in that case, the specificity is also required for drug and ISTD both for alter matrices. So that is definitely required. So the specificity experiment must be designed in a way so for the evaluations of all these three parameters. Now the precision and accuracy of altered QCs. So it is required to process quality control samples in authentic matrix as well as in altered matrix also. Altered means a matrix which are used for the preparations of the calibration curve standard. In the calibration curve, if we prepared with the charcoal treatment, then definitely we need to use QCs with the charcoal treatment. If it is a surrogate matrix, then definitely the QCs must be prepared from the surrogate matrix. Inter and intraday precision and accuracy of altered and authentic matrix QC should be established during the method validation. It is encouraged to use the quality control samples prepared from the altered and authentic matrix during the study sample analysis. So that is also important. Now, recovery and matrix effect experiment. Post-spiked samples are recommended to prepare for the evaluations of recovery experiment at the higher to lower QC level. In case of matrix effect, aqueous or neat samples to be prepared for the evaluations of ISTD normalized matrix factor. In both the experiment, area or response obtained from the endogenous content present into the respective matrices should be separately measured and taken into the consideration for the accurate determination of the percentage of recovery and matrix factor assessment, because it is very much important, because the matrix which we are using for the recovery experiment, it's already contain a few content of endogenous, so that we need to separately measure and to be considered in account for final percentage and calculation of recovery. Now the fourth one is the dilution integrity. It is very much important to identify or select the appropriate matrix to present this for sample dilutions when molecule is endogenously present. It should be analyte-free matrix and normally preferred the same matrix which are used for the preparations of the calibration curve standards. As common point, response of IS should be closely monitored in altered matrix versus the authentic matrix and must be within the acceptance limit. So as mentioned earlier, when we are diluting the samples, the response of ISTD must be monitored. Is there any variation or not? So that's why the dilution integrity experiment is also very much important for such molecules. The stability experiment in authentic and the altered matrix. So as mentioned earlier, definitely stability must be performed in both the matrix because it is very much required. The first is stability assessment, bench stop stability, dry extract stability, stability of extract in reconstitution solutions, long-term stability in matrix at the storage temperature, maybe it's a -20 or -50, whatever it is. So all stability must be performed in altered and unaltered matrix during the method validation and should be reported in the method validation report also. Now we are talking about this US FDA and ICH guidance. So endogenous compound details are already mentioned, and these all details we have covered in our presentations also. The last one is the parallelism should be evaluated for assay for endogenous compound. This one is the newly introduced during the 2018 revision. And in ICH M10 also it is mentioned. Now what definition says in the guidance, we'll see here. The parallelism demonstrates that the serially diluted incurred sample response curve is parallel to the calibration curve. Parallelism is a performance characteristic that can detect potential matrix effect and interactions between the critical reagent in an assay. Now, what ICH M10 says about parallelism? Parallelism should be evaluated in the surrogate matrix and a surrogate analyte. Approaches by mean of the standard addition approach, spike recovery or dilutions linearity. Means this parallelism must be evaluated during the development or maybe in validation with the serially dilutions of the higher concentration, and that we have to compare with the surrogate matrix, or we can say the alter matrix. Now, how to establish the parallelism? So there are some insights are available in the guidance document, some articles, some literatures are showing some details. So that these are some of the extract from that literatures. The sample obtained by diluted samples containing an analyte at the higher concentration. This sample analyzed using a calibration curve generated by a surrogate matrix spiking with the reference standard. Obtained value converted by respective dilution factor are compared to each other. Now the parallelism between a true analyte and a surrogate reference standard may also be evaluated. If the analyte is also used as a surrogate, it is very exceptional cases, but not. But it is also mentioned in the guidance. Even though the parallelism must be established, in short, the parallelism should be designed with the higher concentration sample, that higher concentration sample diluted with the surrogate matrix serially. Now this diluted sample quantitate against the surrogate matrix CC, and whatever the concentrations obtained, that concentration should be evaluated with respect to dilution factor and should be compared. If it is matching each other, then your parallelism is established. So that details are mentioned in the literatures and articles. Now, few of the limitations are there. The experiment design and procedure is not clearly defined in any guidance document, and the acceptance criteria not clearly defined. So further to be explored how to perform this parallelism, but these are few basic details mentioned that we can try and we can perform during the method validation also. Now I going to describe some case studies. These are the developed and validated method in Veeda. I'm taking the four example, different approaches. One is the CC prepared from the charcoal treatment. One is the CC prepared from the photosynthesis. One is the CC prepared from the blank subtractions, and one is the CC prepared by using the surrogate matrix. So this is the first example of estrone and estradiol, where the CC is prepared by the stripping with the charcoal. So QC prepared from alter matrix and authentic matrix by K3DTA plasma. The blank samples, the baseline corrected from this baseline level at -48, -24, and 0 hours. Considering the sensitivity requirement, 5500 equipment is used to get the higher sensitivity. You can see the LLOQ level is 3 picogram and 1 picogram for unconjugated estrone and the estradiol respectively. So that method was developed, validated on 5500, and absolutely apply on the study sample analysis also. And the ISR acceptance for this method is more than 96%, which gives the best result. Now, the second example is the phytonadione. As I explained during my presentation, you just see here we are using the photodegradation, sunlight exposure to plasma. So that we are using for the preparation of CC. QC prepared by using the both alter matrix and authentic matrix. Here two isomers are there. One is the E isomer, that is the trans configurations, and the Z isomer is the cis configuration. So both isomers are quantitated during the development and validation. And from the specific analytical column, we achieve the separations of these both isomers. Now, one of the changes done during the development is a addition, standard addition for the cis phytonadione, because in cis, the endogenous level of cis phytonadione is quite lower. So to maintain and to reduce any bias during the quantitations, additionally we add cis phytonadione in the matrix for the quantitations, and accordingly we do the quantitation. So these types of approaches we applied in phytonadione, and absolutely as per my earlier discussion, we applied this method to our study sample analysis. We did there is a many studies by using these methods, and you see the ISR acceptance is more than 96% for both the isomers. So this shows the successful implementation of this method also. Now the third one is the serum total iron, TOI, and transferrin bound iron. So this is the third approach where we are implemented the blank subtraction method. Yes, here we are applying the blank subtractions. The blank samples are analyzed along with the CC, and the mean value of the blank is subtracted from the CC, and then zero through calibration curve was achieved. So that's why it is used. Why this approach used? Because during the development we see The stripped serum or plasma or diluted serum or surrogate matrix, the response of ISPDase significantly varies, which may raise the matrix factor concerns. So because of that, these approaches are not useful. And therefore, as an alternative approach, similar matrix has been used for the preparations of calibration curve standards. And finally, the endogenous level blank subtraction approach is applied for the final regulation, provided the robust and the reproducible results without any significant variation in the ISD response. Again, it is applied to the many studies, and the ISR acceptance more than 93% for TOI and more than 90% for TBI. The last one is the again the premier method of Veeda, that's the amino acid. This is the one of the premium method where the 20 amino acid is measured in one single method, and these all 20 amino acids are endogenously present. So here we are using the surrogate matrix as a molecule water for the preparations and the generation of the calibration curve, because it is used for the better results and results. Sorry, just a minute. Okay, so this method again we have used for the estimation of amino acids. Here we are using the altered QCs and unaltered QCs, both the plasma QCs and the QCs prepared from the molecule. Now there are some challenges observed during the method development where because all 20 amino acids have in some different endogenous level. So how to generate the calibration curve and everything. So we are define those amino acids in a group wise, the group 1, group 2, group 3, group 4, and group 5. Accordingly, the linearity range has been set as mentioned in this slide and this table. So secondary is the due to the lower molecular weight, difficult to get the stable ionization fragmentation. Therefore, derivatized with the derivatization reagent, which is the 6-aminoquinoline and hydroxy 6-enamide carbamate. This derivatization method is used for the final MRM transitions and everything. Now also the chromatography is one of the challenge, how to separate and set the chromatography for all 20 amino acids. So the chromatographic separation was achieved by using the specific analytical column with the gradient illusion of mobile phase, and overall runtime is about 36 minutes for all 20 amino acids and its respective deuterated IS studies. So with these changes and challenges, we validate that amino acid method, and also this method is applied for the study sample analysis, and the ISR acceptance more than 92%, average of all 20 amino acids. So this is again a very big success for Veeda to develop, validate, and implement this method to the study sample analysis. Now, this is some again very good molecules that we have a validated method with us. This is the estrone, estradiol, alpha-1 acid glycoprotein AAG, amino acid, cis and trans phytonadiene, levothyroxine, hydrocortisone, thymine, iron sucrose, potassium chloride, levocarnitine. We did development, validations, and apply to the study sample analysis for all these molecules, and we'll get the excellent results. So these are our very good and very nicely developed and validated molecules of Veeda's method library. So these are a few references. These references and article I referred during preparation of these presentations, and almost whatever the work mentioned in these presentations is our own Veeda's work that we develop validate methods in our premises, and those case studies are only mentioned in these presentations. So with this presentation, now I stop myself, and I will request all of you to, you can raise your questions and answers. So our panelists can answer your questions, and we'll do some discussion on it. Mr. Hamel, over to you for further questions and sessions.
Yes, Thank you, Dr. Pritesh, for explanation of entire presentations regarding endogenous molecule applicable to BA studies and nicely covering key topics like how diurnal variation to be accountable for endogenous molecules and its resolution by applying different approaches, as well as selection of analyte free matrix for preparation of calibration curve standards and quantitation of endogenous drug, methodization requirement with respect to regulatory guidance, specifically stability studies and insights for parallelism. Now, moving on, we will be proceeding ahead towards our panel discussion round, where we are going to answer some of really interesting questions posed by our audience. Among of that, the first question is for Ms. Swati Gutikar. But before asking the questions, I would give a little introductions of her. Ms. Swati Gutikar is leading as a head of the department at Bioanalytical Research Department, Veeda, Ahmedabad. So the question is that photodegradation approaches used for alteration of matrix for phytonadione. Can this approach be also applying to other photosensitive endogenous molecules? What would be the process, flow, and limitation? Over to you, Ms. Swati Gutikar. Thank you.
Thank you, Pritesh, and thank you, Hemal, for the introduction. Yes, the photodegradation approach cannot be applied to all the photosensitive endogenous molecules. However, based on the molecule chemistry and certain limitations, this photodegradation can be monitored and then optimized if required further. The first limitation is that when the plasma or serum is exposed to the sunlight or the normal light, it should be rapidly degraded within 4 to 6 hours. And after treatment, the plasma or the serum has to be screened to monitor the degradation for the intended drug. The observed interference after the treatment should be either zero or it should be within the acceptable limit of the targeted LLOQ levels. And the degradation pattern should be monitored separately for the sunlight and for the normal light, because for both the treatments, the outcome is different. So if the degradation pattern is only 40 to 60% in 4 to 6 hours, then this method will not be applicable to the intended endogenous molecule. Because as per stability reports, there are certain methods or majority of the photosensitive molecules, they get degraded within 1 hour, 2 hours, or even sometimes more than a week. So one of the example, if we consider, is levothyroxine, which is an endogenous molecule and also a photosensitive molecule. But we cannot obtain the endogenous free plasma or serum because the degradation is very slow in this case. And if we take the example of acitretin and its metabolite 13-cis-acitretin, the degradation is very significant, and we can observe that when it is exposed to normal light within 6 to 8 hours. So overall, we can say that photosensitive degradation approach can be applied to only some of the photosensitive molecules, and there are certain limitations which are mentioned above. Thanks.
Thank you, Ms. Swati Gutikar, for answering the questions in a very well manner. Now, the second question is for Dr. Vikas Trivedi, and I would like to introduction regarding Dr. Vikas Trivedi. Dr. Vikas Trivedi is leading as a bioanalytical investigator at Bioanalytical Research Department, Veeda, Ahmedabad. And the question is that which method was used for estimation of insulin aspart, ELISA or MSMS? Can you please provide the brief details for this methodology? Over to you, Dr. Vikas Trivedi. Thank you.
Yeah, well, thank you. Thank you for your kind word. Just I would like to say that we have applied ELISA technique method for the estimation of insulin as part in the serum sample by using commercially available human insulin kit. So this actually this kit is not distinguish the human endogenous level and the as part exogenous insulin as part level. Therefore, we had to measure the C-peptide level as a biomarker for the exogenous insulin as part. So regarding the insulin ELISA method, we have used a stripped serum for the preparation of CC by using the test formulation, and QC samples were prepared in test formulation as well as the reference formulation to demonstrate the biosimilarity of the product as well as the performance of the assay. Regarding the ELISA C-peptide method, we have used the stripped serum for the preparation of CC by using a working standard, and QC samples were prepared in a stripped serum as well as in the authenticated matrix by using the working standards. And this entire method validation was performed as per the regulatory guidance requirement and by using the duplicate standards to demonstrate the performance of our assay. And the correlation of endogenous insulin human with respect to the C-peptide were performed by using ONS method equation, and this is well explained by the Dr. Pritesh Contractor during the presentation. And this validated method we use for the analysis of the study samples for the performance of bioequivalence study, and more than 3,000 samples we analyzed, and reproducibility of the method we checked by performing the ISR, and the 95% of the ISR samples were found well within the acceptance criteria. So I hope I address all the major consensus of the method. So thank you. Over to you, Emil.
Thank you, Dr. Vikas Trivedi. Very well answered the questions regarding methodology of estimation of insulin aspart. Now, the third question is for Dr. Ajay Gupta. But before asking the questions, I would like to give a little introduction of him. Dr. Ajay Gupta is a leading bioanalytical investigator at Bioanalytical Research Department, Veeda, Ahmedabad. The question is that how to select or fix the required LLOQ for endogenous molecule, specifically when we are targeting blank subtraction method for final regression of
Thank you, Emil, for giving me an opportunity to answer these questions. In general, for endogenous molecule, we use stripped matrix or surrogate matrix for the preparation of calibration curve standards. And the LLOQ should be fixed in such a way that it can measure the endogenous level in designated matrix appropriately. We can fix the LLOQ around 30% of anticipated endogenous level of intended drug in biological matrices, or as per study or method specific requirement based on the experimental trials. However, while using the blank subtraction method for the final regression of CC standard, LLOQ should be kept at least 50% or more of anticipated endogenous level. This approach can minimize the possible variations observed at LLOQ level during the blank subtraction process. Let's take an example of iron formulation. The anticipated endogenous level of iron is about 0.7 to 1.4 microgram per ml. So mean value would be about 1.0 microgram per ml in human serum samples. Hence, it is suggested to keep the LLOQ around 0.5 microgram per ml. So the whole concentration at LLOQ level would be about 1.5 microgram per ml, and the area observed at the blank sample, which is about for 1 microgram per ml, is subtracted from the LLOQ to ULLOQ concentration for final regression of CC standard. So we have to keep the LLOQ around about 50%. If we are keeping the LLOQ less than that, but considering the example of iron formulation, we keep the LLOQ like 0.2 microgram or 0.3 microgram, then this may impact significantly at lower concentration due to significant variation in response at LLOQ level. So selection of LLOQ is important while we are targeting blank subtraction method. Thank you. Over to you, Hemal.
Thank you, Dr. Ajay Gupta, for answering the relevant questions of LLOQ requirement for endogenous molecule. My fourth question is for Mr. Jesuk Rathod, and I would like to introduce a little introduction of him. Mr. Jesuk Rathod is a leading as a senior group leader at Bioanalytical Research Department, Veeda, Ahmedabad. And the question is that it is required to use quality controls prepared from strip or surrogate matrix during the study sample analysis. Which matrix to be used for sample dilution during the study sample analysis? Over to you, Mr. Desuk Rathod. Thank you.
Thank you, Hemal, for given opportunity to answer this question. Now can I say it is the expected answer from the bioanalyst who is involved during the sample analysis of endogenous molecule. Now move to the question regarding the uses of altered QC. None of the regulatory guidance is clearly indicate or suggest to use the altered QC sample during the study sample analysis of endogenous molecule. In one of the presentation given by the USFDA in last year, they also do not mention clearly to use the altered matrix during the study sample analysis. However, it is recommended to use the altered matrix QC sample as well as authenticate matrix QC sample during the study sample analysis. Now move to the second question. Regarding the sample dilution, it is recommended to use the altered matrix to dilute the study samples. This altered matrix is the same which is used for the preparation of calibration curve standards. In rare case where the authenticate matrix was used for the preparation of CC standard, there we need to dilute the study samples by using this authenticate matrix only. But in this case, we have also considered the endogenous concentration of this matrix while calculating the dilution final concentration. I hope I answered this question in a satisfactory manner. Thank you. Over to you, Hemal.
Thank you, Mr. Desuk Rathod, for the depth explanations regarding requirement of matrix for the sample dilution during the study sample analysis. Thank you all the panelists for your active participation in this panel discussions and nicely answering to most of our audience questions. I would like to take a pause over here. We understand this session has kept us all well engaged throughout, but due to time constraint, we would not be able to take any further question at this point of time. But please be rest assured that we will be sending the answer to each and every questions of yours via emails. Also, we will be sending across the recorded link of this webinar on all the registered email IDs. Lastly, I myself, Hemal Kurani, on behalf of Veeda Clinical Research, would like to sincerely thank you all of being here today and taking out the time to be a part of this webinar. I would also like to extend our gratitude to Dr. Pritesh Contractor for being such a wonderful presenter and giving us all great learning today. We at Veeda Clinical Research earnestly look forward to host you all again. Till then, take care and stay safe, everyone. Thank you very much.